Hello, welcome friends. This is Namdev Mahale. I will present the topic today called as phases in mitosis. Learning outcomes. First, learners will know about basic steps in mitosis and can learn it step by step. They can easily understand the process of cell division and cell duplication phenomenon. Learners will able to differentiate mitosis in plants and animal cells. Learners will able to differentiate between mitosis and meiosis. Let's move further. Contents of the video. First, we will look into the introduction, then stages or various phases of mitosis. Summary will be discussed at the last of the video. So, in introduction, basically we will see the definition of mitosis. So mitosis is a process in which eukaryotic cell nucleus splits into two, followed by division of parent cell into two daughter cells. The word mitosis means threads, and it refers to the thread-like appearance of the chromosomes as the cell appears to divide. Mitosis is very basic and fundamental process of cell division which occurs in an organism which will which we may eukaryotic or prokaryotic organism. It is slightly different in animal cell than in plant cell. The phenomenon is different because in plant cell there is a absence of centrosomes, and in animal cells there is a presence of centrosomes, which leads to the radiating centriole spindle fibers. So at very first, the appearance of specialized network in microtubule during mitosis, these tubules, collectively known as spindle or spindle fibers, which extends from structures called centrosomes, with one centrosome located at each of the opposite ends or poles of the cell. So, friends, you will learn about this uh, sentence, that is the cell we are discussing over here is the animal cell. As mitosis progresses, the microtubules attach to the chromosomes which have already duplicated their DNA and aligned across the center of the cell. The spindle tubules then shorten and move towards the poles of the cells. As as they move, they pull the one copy of each chromosome with them to opposite poles of the cell. This process ensures that each daughter cell will receive one exact copy of the chromosome in cell DNA. We will discuss here the phases of mitosis. First phase, the interphase. During this interphase, uh, various changes happens in the cell, which includes increasing in the cell size. So, size of the respective cell increases with increase in the DNA replication. Duplication of the centrosomes happens. Then the second stage called as the prophase. In the prophase, various changes occurs. First change is called as replicated chromosome that contains two closely associated sister chromatids will condense. Then the meiotic spindle assembles between two centrosomes which have replicated and moved apart in the outside of nucleus, and nuclear membrane still intact. So nuclear membrane will disappear afterward or in the next stage. These are the centrosomes and which are moving towards the opposite pole. So this is the prophase, prometaphase, sorry. Breakdown of the nuclear envelope is the first change in this stage, and the chromosomes which attach to spindle microtubular tubule via their kinetochores and undergo active movement. So this is the start of metaphase actually. So metaphase, very distinctly known as the chromosomes, they are attached at the metaphasic plate. Here, this stage is very special and distinguished by the arrangement of the chromosomes. The chromosomes are aligned at the equator of the spindle and midway between the spindle poles. Kinetochore microtubules attached sister chromatids to opposite poles of the spindles. So anaphase is the second last phase, which is again very significant because the chromosomes are pulled now towards the opposite pole with the help of kinetochores or spindle fibers, which are attached with the centrosome. The sister chromatid synchronously separate to form two daughter chromosomes, and each is pulled slowly towards the spindle pole it faces. Kinetochore microtubules get shorter, and the spindle poles also move apart. Both processes uh, contribute to chromosome separation. Now, last, telophase. Telophase is the end of the mitosis, mitotic division. Telophase, the two sets of the daughter chromosomes arrives at the pole of the spindle and decondense. A new nuclear envelope reassembles around each set, completing the formation of two nuclei and marking the end of mitosis. So here these 2 points indicate that the cell will now divide from here to here, and each cell receives equal number of chromosomes. Hence, this mitosis is also called as equational division, and meiosis is also called as reductional division. In meiosis, there are two stages, meiosis 1 and meiosis 2. But in mitosis, there is a single uh, stage, means only meiosis first. We cannot say meiosis first or sorry, mitosis first or mitosis second, but it's simple. In the telophase, two sets of daughter chromosomes arrive at the poles of the spindle and decondense. A new nuclear envelope reassembles. Means nuclear envelope reformation will take place over here and over here. In the last stage of telophase, the division of the cytoplasm begins with the assembly of the contractile ring. As I said earlier, contractile ring will form here, and both the cells uh, will divide from this contractile ring. Last, cytokinesis. Cytokinesis means dividing two cells. Actually, the end of telophase will lead to form a process or to form two cells. The process is known as cytokinesis. Karyokinesis means division of nuclear division, and cytokinesis means division of cytoplasms. So this the phenomenon which leads to the cytoplasm is divided into two by a contractile ring of actin and myosin filament, which pinches the cell into two, create two daughter each with one nucleus. Means here the motor protein actin and myosin will work over here, so that they will lead to divide cells respectively. So this is the practical presentation of slide. Actually, when we do practical, then the diagram looks like this slide. So first is the interface, then in prophase, chromosome slightly become a thicker, and then in metaphase they are aligned on the equator plate, or called as metaphasic plate. In next stage, called as anaphase, the chromosomes are pulled towards the opposite pole, and in the last stage, telophase occurs, where before the cell division, this diagram is showing the formation of two different nuclei at two different places, but without cytoplasmic division. So in the last stage, we will see the cytokinesis. So moving further towards summary. So summary is very simple, that we have already seen, but it is our video part. So it starts with the interphase, prophase, metaphase, anaphase, and telophase. So basically 5 stages are there, and in mitosis, interphase is again very important. In prophase, metaphase, anaphase, and telophase is again very important. Here cytokinesis is not taken, because cytokinesis is the um, last phenomenon which is not involved in the mitosis basically, but we study over here. So this is the summary. Actually, mitosis is interrelated with the cell cycle, where G1S, G2M stages are there. At the M stage, cell divides, and this mitosis takes place at that time. Mitosis is very basic in actively dividing tissues. Like when we consider animal, then if you take tissue called as skin tissue, where you will find mitosis. Mitosis occurs everywhere. With the we will say where gonads are not there, there are mitosis, and where gonads are there, meiosis will take place in that place. So in case of male, human male, where in testes you will find the meiosis, which forms sperms, and in females, uterus is there, and where egg is produced by the process of meiosis. But other than this structure, reproductive structures, everywhere takes mitosis takes place. uh, that is the significance. In plants also, if we consider flower part, androecium and gynoecium, that there reproduction reproductive organs are there, and there you will find meiosis. Other part of that vegetative part of that plant is lead to form uh, various kind of tissue by the process of mitosis. This is our video, and thank you for hearing me.